Hey guys, Kenny here. So I just want to do kind of a quick hit on the basics or history of cell theory and kind of cell discovery so that you guys have something to kind of start with as we go into our next unit on cells and cell structure. Okay? So let's go ahead and jump right in on our essential questions. So number one, what are the three tenets of cell theory? Now, when we say tenets, what we really mean is kind of the key primary principles or concepts that make it up. Number two, what are the characteristics of all cells? There are a couple of organelles in particular I'm looking for here that are the basis for all of, all cells have them. Doesn't matter which one it is. And so we want to make sure you know what are those organelles that are in all of them. And number three, what are the two major cell types or divisions that we kind of separate them out by? And what are their similarities and differences? All right. So let's start by giving you a brief history. We can talk more about this, but the first compound light microscope that we look at is really the one done by Anton van Leeuwenhoek. Now, that's kind of debated whether it was actually invented by him, probably not. It's suggested that it was invented by a gentleman by the name of Zacharias Janssen, but I include van Leeuwenhoek here because of his contributions to the study of bacteria and microorganisms. But he was able to see more things and share his knowledge with a lot of different people, and that's really where a lot of this started. The cell was discovered by Robert Hooke in 1665 when he was actually looking at cork cells. He examined some very thin slices of cork and saw a whole bunch of tiny little pores that he remarked that looked like walled compartments that monks used to live in. Because of this association, Hooke called them cells, and that's the name they still bear. There's no other reason that they're called cells other than that this is what Robert Hooke called them. Okay. All right. So, and there's a whole series of interactions with a whole bunch of different people that lead to all of this happening. But these are kind of the key people that we want to take a look at. The observations of Matthias Schleiden working with plants and Theodor Schwann working with animals leads us to the realization that all living things are made up of cells. But they tended to argue about how cells came about, whether they came from other pre-existing cells or they just appeared through a process of something like spontaneous generation. The gentleman named Rudolf Virchow knew the work of Francisco Redi on spontaneous generation and many others, and extended this to state that the only source for a living cell was to come from another living cell. And while Virchow is given most of the credit most of the time, it is quite possible that Virchow stole this idea from another scientist, Robert Remak, but it's unclear. Okay. But if we take the work of all of these individuals together, we end up coming up with what we call cell theory. And the cell theory is a widely accepted explanation of the relationship between cells and living things. The cell theory states three things. One, all living things are composed of cells. Two, the cell is the basic unit of structure and function for living organisms. And three, all cells arise from pre-existing cells. And this is something you probably want to make sure that you know so that you're ready for it when it comes up on the test. Okay, so we have a cell. What do we know about cells? Well, that's what we're going to go ahead and focus on in this unit. But we want to understand that there are some basic things that are found in all cells. All cells have the following parts. One, the cell membrane, at least one of them, that separates the cell itself from the external environment. Two, inside that cell membrane is the cytoplasm, some sort of fluid where all of these life activities take place. And three, they have to have ribosomes because ribosomes make proteins, and proteins do all of these key jobs throughout the cell. So, how do we know this? How do we come up with cell theory? The material that we now know is kind of the basis for what we're going to be covering was only possible because of the tools of investigation that they had. And those tools were primarily microscopes in this case. So there are two major categories of microscopes. We have the light microscope and we have the electron microscope. Now the electron microscope is definitely much newer, and it is a powerful tool. And there are two main types of electron microscopes, which we'll go over here in a little bit, the TEM and the SEM. Let's start out by taking a look at the light microscope. Now this is very similar to what you're going to be using here at Willamette High School. Compound microscope is what you have in all of your biology classes, and the stereoscope, something like what you see on the right, is what we use typically in like a geology or a botany, maybe in forensics, something like that. Typically for a little larger, it doesn't zoom in quite as much, and the compound microscope typically is going to look at very thin slices and zoom in a fair amount. The next group are those electron microscopes. We have our SEM, which stands for scanning electron microscope. Okay, it's actually going to go ahead and shoot electrons at the object, and then it's going to read based on that where they bounce to, and then build a three-dimensional picture from that. And then you have a TEM, or transmission electron microscope, which tries to throw electrons at the object, and then whatever passes through or doesn't pass through is going to go ahead and produce kind of a silhouette picture of the cell. Let's start out by taking a look at TEM, because it's actually an older type of microscope than the SEM. In TEM imaging, the image is created by measuring the light that is transmitted through the specimen. So if you have a dark spot, it's because it hit something. So it didn't go through to the actual other side of the microscope. And so you end up with pictures like this, but that can tell you a lot about the internal structure, and we're able to zoom in and see a lot more than we previously could. This was expanded to produce even more more powerful images when we switched over to the SEM microscope. Now, the SEM, as I mentioned, is you place the object inside the chamber, and then the electrons are thrown at it, and then they are reflected and dislodge electrons from the specimen, and as such we can go ahead and see fairly meticulous three-dimensional images. Does anybody want to guess what these are pictures of? Mm-hmm. So right here, what we see here on the left, this is actually going to be the mouthpiece of a fly. Okay, and the top right here, this is actually a mattress mite. It eats off your dead skin cells. Don't confuse it with a bed bug. They're not the same thing. And then down here in the lower right, this is actually viruses. Okay, so very powerful tool to see some pretty amazing three-dimensional images of the objects that we're going to go ahead and take a look at. But because of these powerful tools, we're able to go ahead and understand much more about organisms, microorganisms, and even cells. And so because of this, we've been able to separate cells into two categories. Those categories are prokaryote. Okay, these are your simplest cells. They're going to go ahead and not have a nucleus, and they're typically missing a lot of the organelles. And they do contain cytoplasm, cell membrane, and ribosomes. Remember, all cells do. And this includes things like bacteria, your E. coli, for example. The other category is the eukaryote. Okay? Now, that prefix EU means good or normal, and so we call these eukaryotes because we're talking about, well, this is the kind of cell we have. Versus pro means before, like prototype. So these are the cells that came before we did. So eukaryotic cells contain many different organelles, like many. So we're going to deal with a whole list of them that we'll go over next class and talk about all the different functions they do as they specialize and make us more efficient in terms of our cellular level processes. And this includes things with membrane-bound or membranes, okay, bound in membranes. So we start thinking about things like Golgi apparatus and mitochondria and chloroplasts, just to name a few. And we subdivide the eukaryotic class into 4 different groups, but primarily we're going to talk about 2. We're going to talk about plants versus animals because there are noticeable differences. You could also focus on fungus and protists, but we're dealing with the same basic pieces. So if we cover plants and animals, you'll get to cover all of the organelles that we're going to go ahead and discuss. So here is a photo and a diagram showing a prokaryotic cell. You can see that we definitely have ribosomes. In this case, we also have a cell wall outside of that plasma membrane that's going to help create that outer barrier. And then there's no nucleus, but we do have a nucleoid region where the DNA is floating around inside of the cell. Then we have our two eukaryotic types of cells. This first one is going to be an animal cell. Go ahead and look for those same pieces here. I can see the plasma membrane. I can see the ribosomes. And I don't think they've labeled it, but you've got your cytoplasm in there as well as a whole bunch of other organelles that we'll talk about next class. And then here we've got our plant. Again, take a look for it. Let's see if we can find them. There's your ribosomes. There's your plasma membrane. And there is a fluid inside. Now notice that this one has a cell wall, whereas the animal cell did not. And this one has a much larger central vacuole, where you didn't see that in the animal as well. Okay, so that's your brief introduction, and we'll go over a lot more of these organelles in our next class. So until then, stay healthy, stay safe, and we'll talk to you soon. So long.